Okay, thank you, Ned. Thanks for the introduction, and particularly thanks for inviting me out here. It's great to be here. I hear that it's always like this in March in Boston. So thanks for being inside instead of enjoying the weather. And I'll try my best to keep everybody away from burrito food coma here. So let me just start off by saying this is how I see the world. Okay, and the key thing I want to point out here is that most of the biological diversity on Earth is microbial. In other words, most of the biological diversity is represented by single-celled organisms. So this is a tree of life as it currently stands, and it stands for revision, but this is currently where we have bacteria, archaea, and eukaryotes. And everything that's in pink are microbial lineages, so single-celled lineages. So the majority of the diversity is microbial, and that's where you are. So I think it's important for our hubris to remind ourselves that we are just small branches on this enormous tree, and most of what biologists study, the animals and plants, are still just a relatively small sliver of all the biological diversity that's out there. And this is true just from an evolutionary perspective, but also from a physiological perspective. As far as I'm concerned, plants are oxygenic photosynthesizers, and we're aerobic respirers. So we basically use oxygen as an electron acceptor and get our energy from food. But in microbes, you have a full diversity of different physiologies, organisms that can eat iron and get their energy from it, organisms that eat arsenic, and so on and so forth. The other thing I want to point out, and most of you probably know this, is that microbes are ubiquitous and abundant. This is good news if you're a microbial ecologist, bad news if you're a germaphobe. Right? So any environment we look at, so we're having a minimum of at least 1 billion cells per gram of soil, millions of bacterial cells per meter cube of outdoor air. They're abundant on leaf surfaces, on the dollar bills that you touch on a regular basis. And you don't even need to go to such exotic environments like soil or leaf surfaces. We can also look inside our own bodies. Okay, so we have about 10 trillion human cells in our body and about 100 trillion microbial cells in the human body. And this is true no matter which body habitat you're looking at. If you're looking at the mouth, the skin surface, your large intestine, the majority of these cells, the bacterial cells, are actually in our large intestine. So about 10% of, only about 10% of the cells in the human body are actually human cells. And of course, we have a difference in scale. Bacterial cells are typically quite a bit smaller, but you're still carrying around at least 10 kilograms of bacterial biomass on you, on average. And that number varies, but there's a lot of bacterial biomass inside you. And what I want to point out, and sort of the theme for this talk, is that the vast majority of the microbial diversity that's out there remains undescribed. And that's true for the bugs in soil, the bugs on leaf surfaces, and those inside your own body. We know they're there, but we know surprisingly little about what they're doing, how they make a living, and so forth. And I think the best summary of the state of microbial ecology came from, we'll call him the patron saint of microbial ecology. That was Don Rumsfeld. And he says, this is the famous quote, of course, he says, there are things we know. We also know there are known unknowns. That is to say, we know there are some things we do not know, but there are also unknown unknowns. The ones we don't know, we don't know. Okay, that's a mouthful. But the point I want to make is we know in microbial ecology that the unknown unknowns and the known unknowns far outweigh the number of known knowns. And you can see this if you just look in the literature. Literally every week there's some huge discovery of microbial diversity or a new understanding of the role of microbes in our health or the health of animals and plants. So it's really an exciting time to be in the field of microbial ecology. And the reason we know so little about microbial diversity is because the methods that have been used for the past 300 years of microbiology miss a large portion of the microbial diversity that exists out there. So typically what microbiologists have done since the dawn, since Louis Pasteur in the 18th century, is they basically take a sample from your mouth or the soil or what have you, and they plate it out on a Petri plate, and then you identify the different types of bacteria that grow on that plate. And this is, of course, what you do when you go in for a strep test, for example. The exact same thing happens. And fortunately, most pathogens we can culture this way, so we can identify whether you have a particular bacterial infection and so forth. But it turns out, at least in environmental samples, more than 99% of the bacteria are resistant to culturing. So we can't get them to grow in this sort of artificial lab-based environment. But we can start using DNA analyses to survey bacterial diversity. What this means is we can take a scoop of soil, or we can take a wash of a leaf wash and what have you, and extract the bacterial DNA from that sample and use that DNA sequence information to tell us who's there. So we don't have to rely on these sorts of methods. And there's really been a revolution in the field of microbial ecology just in the past 10 years. And a lot of this is spurred by methodological advances. So it's now a lot easier to get this DNA information. So we can start collecting hundreds of samples and looking and see what types of bacteria are in there. And part of this is spurred on by just the fact that DNA sequencing is dropping dramatically in cost. So this is just time on the x-axis here, and this is how much it costs to sequence a human genome. So the technology is exactly the same. DNA is DNA is DNA. Except instead of looking at human genome, we're actually looking at specific bacterial genes to identify what types of bacteria are there. And if you talk to various venture capitalists, this is way out of date, and the number is probably around $1,000 per human genome. But we can take advantage of these advances in DNA sequencing to explore new territory and look at microbial diversity in all sorts of environments. I should also point out that here's Moore's law. So this is the number of processors you can fit on a chip. So basically we can generate data, DNA data, a lot faster than we can actually analyze it because the speed of acquiring DNA sequences is so high. So what does this mean in terms of microbial ecology? So in 2006, when I started at University of Colorado, if you gave me $10,000, this is what I could do. I could take a sample, whether we're talking about soil or a skin sample or what have you, and I could pick out 100 individual bacteria from that sample and identify them based on the DNA sequences. And I could do this in about 25 samples. Okay, now, for the same amount of money, I can pick out 10,000 individual bacteria from each sample and identify, okay, we have this type of bacteria there, and I can do this for each of 500 samples. So what this means is we can address questions that even a few years ago were like, well, that's a great question, but we we don't have enough sequencing capacity to do it. And now it's pretty straightforward to analyze hundreds of samples. And analyze those samples in depth so we can really get a good idea of what sort of diversity is in those samples. So we can do projects like this. Okay, so I don't like to travel too far. So here's my office, right about here. So this is no Harvard Arboretum, okay, but at the University of Colorado, there's lots of tree species that were planted 50 to 100 years ago all around campus. And we can go in and sample bacteria off the leaves of these different tree species. And we ended up with about 350 samples. We took replicate samples from the different tree species and so forth. We can essentially ask the question, well, what types of bacteria live on the leaves of these trees? Where I'm not going to discuss is we actually see there's a huge amount of interspecific variability. So trees harbor unique bacterial communities on their leaves. It's not just you get these bacteria landing on the leaves and whatever happens to be there is there. There seems to be a really strong selection for certain bacterial taxa, depending on the species that you have. And this even extends across, if we sample pine species across Australia, California, and Colorado, we see the exact same types of bacteria on those pines, regardless of where they're located. So a strong degree of species or plant species specificity in terms of the types of bacteria that are found on the leaves. And also the diversity is quite striking. So these are just 3 plant species. These are actually 3 individual samples from individual trees, just to show you what we're working with here. So the x-axis is number of sequences. So it's essentially how many bacterial genomes are we looking at in each sample. So we sampled about 1,000 individual bacteria in each of these samples. The y-axis is number of bacterial species. I put species in quotes here because it's a bit unclear what is a bacterial species. But the fact of the matter is a single leaf, or a single tree, rather, can harbor anywhere, upwards of 400 different bacterial species on the leaves. And notice that these lines aren't flat. So, if we kept sequencing more, we'd probably discover more and more new species. So we're dealing with an enormous amount of bacterial diversity. Okay, so it was too much work to travel 100 yards outside the office and collect samples. So we recruited my postdoc. This is Chris Lauber, who's a postdoc who's worked for quite a number of years. We were like, all right, now that we have these technologies, let's just see what types of bacteria are on your face. So we divided his face up into these different quadrats. And Chris's claim to fame is he's the most sampled man in human microbiome history. Like, we've sampled basically every orifice and looked at all the bacterial diversity on him and generated billions of sequences of information just off his body. But, we can do these sorts of things. And this was not done for scientific purposes, just to see, can we actually pull this off? And this is bacterial diversity on his face. So red means high diversity, blue means low diversity. And first off, I want to point out that we're dealing with a huge amount of diversity here. So individual spots harbor upwards of 300 species of bacteria on his skin. So we have the Amazon rainforest of the mouth and the bridge of the nose here, and far lower diversity on his cheeks and so forth. So, the fact of the matter here is we can start doing these sorts of projects that, even just a few years ago were impossible to carry out. But I don't want to give you the impression that this revolution in microbial ecology is solely determined by methodological advances. And there's been a huge paradigm shift, and this has been occurring over the past 30, 40, 50 years, but it's really sort of been accelerating recently. And it used to be that if you're a microbiologist, you studied germs, right? Bad things. So the idea was that the only good microbe is a dead microbe. So this is equivalent to saying you've got some weed out there, so let's just clear cut the entire forest, right? I mean, it's essentially the same sort of paradigm. And now both medical microbiologists and microbial ecologists in general are starting to recognize that, of course, there are pathogenic microbes. They're no joke. Just take them seriously. But many of the microbes that exist out in the environment, whether we're talking about soil or on leaf surfaces or in our bodies, are actually harmless or may even be beneficial. So it's a much more nuanced view of microbial diversity. And there's increasing recognition that these microbes that live on us or in us can play a critical role in shaping our traits, or what we look like. So you can take genetically identical individuals of plants or of mice and inoculate them with different bacterial and fungal taxa, and you get very different phenotypes, very different growth rates. Most obvious here with the mice. So these are genetically identical mice. The only difference is they've been inoculated with different types of microbes in their gut. So very, very big differences in, obviously, body size here and in plant size. And every week there's new evidence of the critical role that these microbes play in shaping our health and growth rates and so forth. And so I already mentioned that about 10% of the cells in our body are microbial, are human cells, and 90% of them are bacterial cells. But it's even more striking if you look at the genetic diversity. Okay, so we have about 20,000 human genes in our genome, right? And in contrast, we have about 20 million different microbial genes that exist on us and in us. So what this means is that our microbiomes, those microbes that live on us and in us, provide us with a lot of important traits that we did not need to evolve on our own. For example, the ability to eat a lot of what's probably in your burrito. We probably don't have a lot of the enzymes to degrade some of those compounds, but our microbes do, and they can do the digestion for us. Training our immune system, defending us against pathogens, tissue development. A lot of this requires the microbes that live on us and in us for us to be healthy. Okay, so what I want to do the rest of the talk today is give you 3 short examples of how some of our work in a variety of different systems has I think, told us some interesting things about microbial ecology and about microbial diversity in general. And we probably couldn't pick 3 more different study systems. So soil, the near surface atmosphere, and the human body. And I'm going to pick examples that all focused on biogeography. So biogeography generally is just described as why organisms live where they do. And there's a huge history of research on biogeography, and obviously in the plant literature, in animals, birds, and so on and so forth. We know an enormous about their biogeographical patterns. And this is obviously what's been critical and fundamental in a lot of development of ecology and evolutionary biology and so forth. But microbes, you can't see it here, but there's a little question mark. We know surprisingly little about the biogeographical patterns exhibited by microbes, just how they vary across space. And if you've taken biology in the past, let's say 100 years, probably what I'm going to mention today is like, really? You didn't know that? Because a lot of what I'm going to be mentioning is basically research that people knew 150, 200 years ago were for plant and animal taxa. We're asking the very same questions. We're basically saying, all right, well, instead of going to the Andes and collecting plants, or going throughout Indonesia and Africa and collecting insects, let's look at the microbial diversity that exists out there. So let's just first find out what we're working with, and then use that to help us learn something about the microbes and what they're doing in our soil or on our bodies and so forth. So that's my caveat. This is basically 150-year-old science. And let's start with soil. Okay, so the amount of microbial biomass in soil is pretty impressive. So in your average soil, we have anywhere from 400 to 2,000 kilograms of microbial biomass per hectare. A lot of this is bacterial, some of it's fungal, there's other microbes as well, but I'm mainly going to focus on the bacterial diversity patterns in soil. And the diversity, again, is quite astounding. So per meter squared of soil, even if you just take some samples from the lawn out here, we have anywhere from 1,500 to 10,000 bacterial species per meter squared of soil. So enormous amounts of diversity that we're working with here. So our question was, and this was about 5 years ago, the question was, okay, well, why this variance? Why does it vary so much? Which soils harbor more bacterial diversity than other soils? So we essentially cruised around and collected soils from all over the place. So this is just a map of at least the initial sampling. We tried to get soil from as many different ecosystem types as we possibly could. Everything from Arctic tundra to tropical rainforest in Peru to deserts to prairies and so on and so forth. And the idea was, well, let's just see what's out there. Let's just collect soils from all these places, and these were all collected during the growing season, and let's just see what sort of diversity patterns emerge. And our hypothesis was this. So this is a map of mammal diversity across North and South America. And this isn't true for all plant and animal taxa, but most plant and animal taxa typically have highest diversity near the equator. Okay? And again, it's not true for all taxa, plant and animal taxa, but many of them. And this has been well studied. We've known this since 1800s, basically, that diversity tends to peak. For many taxa, it tends to peak near the equator. So this is what we hypothesized. And it turns out you can actually predict the diversity of many plant and animal taxa by measuring how warm and wet it is. So basically, in places that have high potential evapotranspiration, so places that are warm and wet, you tend to see high diversity of birds, of mammals, of amphibians, and reptiles. Okay, so we thought, well, we should be able to predict bacterial diversity in soil from this same parameter that's been relatively well studied for plant and animal taxa. And we can't. Okay, so the y-axis here is phylogenetic diversity. So basically, higher means higher diversity. It's just the number of different lineages in a given sample. And mean annual temperature doesn't predict it. Latitude doesn't predict it. How warm and wet it is doesn't predict it either. So all these parameters that we know predict the diversity of most plant and animal taxa do not predict bacterial diversity. Instead, it appears to be soil pH. So soil pH is a surprisingly good predictor of bacterial diversity, where diversity peaks in soils that are near neutral. And you see this on a global scale, and you also see this on a local scale. If you take pH gradients that vary from like pH 4 to pH 9, you can see this exact same pattern, even if you're controlling for climate and plant type and so forth. You see these exact same patterns replicated both on local scale and on continental scale. So, and we think the reason for this is most bacteria, the intracellular pH, the pH inside their cells, is always close to neutral. So it must just be some sort of stress to be either far above neutral or in very acidic conditions. So let's zoom in on this graph here. So this is what was most surprising. So the highest diversity soils are actually prairie soils. You see very high levels of bacterial diversity. This is the area with at least plant diversity, or at least tree diversity, is probably the highest we know of, at least in North and South America. We actually have the lowest levels of bacterial diversity in these soils. And again, it's probably because these are very acidic. They're very highly weathered soils, very acidic. And so since then, so that was based on about 110 soils. And since then, we've gone around the world, basically, or we've had soils shipped to us around the world. So we can start putting together a map of bacterial diversity across the world. And a lot of this is driven by pH, but not solely by pH. It's the best predictor, but it's not the only predictor. There's other things that also shape bacterial diversity. But this is what we think is sort of the map of soil bacterial diversity at present. And this is a number of genera, so red means higher diversity, blue, purples mean lower diversity. And let's compare this to a map of oh, it didn't show up. Okay, sorry, there was some, but basically it's supposed to show that in the lower latitudes, so near the equator, we see the highest levels of plant diversity. And in contrast, we actually see the opposite if we're looking at soil bacterial diversity. The tropics actually have the lowest levels of diversity in their soils. So we're seeing this huge disconnect between the biogeographical patterns exhibited by plants and that exhibited by soil microbes. Okay, so let's shift to another environment entirely. And again, we're asking the same sort of question, and this time looking at the atmosphere, of how does microbial diversity vary across space? And why do we care about the bugs in the atmosphere? Well, every time you're outside, like on a beautiful day like today, you're inhaling anywhere from 10,000 to 10 million bacterial cells per hour. And this is just bacterial cells, when we're not even counting the pollens and the fungi and all these other things that you're breathing in. And of course, we know anyone who suffers from allergies knows that some of these are not good. Probably the minority of them cause health problems. The vast majority are probably harmless in the atmosphere. But we know, for example, that in the United States, at least, there's 10 million individuals with allergenic asthma or allergic asthma. And there's increasing evidence that a lot of allergenic reactions are actually triggered by bacteria, not by fungi and pollen. There, of course, can also be allergens, but bacteria can as well. And it hasn't been well studied because, of course, the methods we use to study bacteria, growing them on the plates, don't work for the vast majority of bacteria that are out there, particularly in the atmosphere. So it's hard to test if they're allergenic or if they cause allergies because we can't grow them and do these experiments with them. But there's increasing evidence a lot of people, especially seasonal allergies, triggered by bacteria in the atmosphere. And of course, there are pathogens, for example, fire blight, that's caused by bacteria that are readily dispersed through the atmosphere. Okay, so and I also want to point out that there's a long history of research on microorganisms that live in the atmosphere. And this goes all the way back to the days of Louis Pasteur in the late 18th century. And even more recently, this is work by Fred Meyer and his co-author, who you may have heard about, Charles Lindbergh. So this is Charles Lindbergh of the transatlantic flight fame. And what he did, he was a co-author in this paper because essentially he flew across the Greenland ice cap and stuck a filter out the window of his plane. Like, I'm not joking, like, he literally stuck it out there. It must have been pretty cold. And basically they brought these filters back and they said, all right, well, what's in there? So they see some fungi, and they see some pollen, they see some bacteria, and they basically say, let's just describe the diversity of microbes that are out there over the Greenland ice sheet. And this is basically where the study of airborne microbiology stood until recently. It was either using morphological identification or culturing them on a plate. And again, we remember that the majority of microbial diversity that's out there doesn't grow on a plate. So we were missing a lot of the bugs that were blowing around, that we're breathing in every day we go outside. Okay, so a study we wanted to do was, all right, well, let's just take samples from a handful of locations. And it's actually really hard to get samples that are not contaminated, where you know what you're sampling is actually bacteria in the atmosphere, not somebody touching a filter and so forth. And this is 4 sites: Detroit, Chicago, Cleveland, Mayville, Wisconsin, a smaller town here. And we collected samples once a week during the winter months and once a week during the summer months. And just for the record, I love all these cities. You'll see why I'm saying this in a second. But if you do have relatives in the upper Midwest and you want an excuse to not visit them during the holiday months, I'm about to give you one. In case the weather isn't enough of an excuse. Okay, so what I'm showing here is, I apologize for the messy graph. So each point here, these are identical. Each point here represents an air sample, or a bacterial community from a given air sample. Points that are close together represent samples with similar bacterial communities. Points that are far apart represent air samples with very distinct bacterial communities. Okay, so what I have here is, they're colored just by where they were collected. And as you can see, there's no big patterns. But we do see a strong seasonal pattern. So in other words, those bacterial communities that are blowing around, regardless of the city in the summer, are very distinct from the types of bacterial communities we see in the air in the wintertime. Okay, so this is interesting. So we're like, what's going on here? And then I'll spare you the details here, but we started to see some bacterial taxa showing up in these samples that we didn't expect. And in particular, we started to see a lot of bacterial taxa that we thought were only restricted to fecal samples. And these are bacterial taxa that we don't see anywhere. We don't even see them in sewage treatment plants. They're very rare in sewage treatment plants. But we started seeing groups, phyla of bacteria, or members within these phyla of Bacteroidetes and Firmicutes and Fusobacteria. And again, don't worry about the names. These are taxa that, as far as we knew, we didn't find them anywhere else except for fecal samples or in the gut. And in particular, we were really surprised to see Fusobacteria. So Fusobacteria is a group of bacteria that we have a few of them in our mouth, but they're very rare. But they're very, very abundant in carnivores. So we're like, what's going on here? And fortunately, we had done work in a wide range of other systems, so we could start quantitatively comparing our results that we found in the bacterial communities we found in the air samples to those found in potential source environments. And again, all you need to know is each point here represents a unique bacterial community or bacterial community in a given sample. Points that are close together represent samples with similar bacterial communities. That's all you need to know. So these are the air samples up here, and these aqua, these are the wintertime air samples. So we're starting to see them looking like fecal, those bacteria we see in feces. And in particular, those we see in dog feces. And it's because we have these unique phyla that we don't, as far as we know, the only source is carnivore feces. And it's highly unlikely that just coyotes and wolves and so forth, I mean, they're going to be there, but not contributing enormous amounts. So what we think is happening we're seeing a huge source of basically aerosolized dog shit entering the atmosphere. And it's very difficult to confirm this using alternative methods, but everything points to dog feces as an important source of bacteria in the winter months at these sites. And so, we can use these bacterial communities to do essentially source tracking, and this is what it looks like. We're seeing the soil is not a particularly huge source of bacteria at this site. We do see in Colorado, where it's quite a bit drier, we do see a lot of soil bacteria in the atmosphere. And in the summer, the majority of the bacteria that we find in the atmosphere is actually coming off of leaf surfaces. They're probably just getting whisked off of leaves, or as grass is being cut, the bacteria are being aerosolized and so forth. And in the wintertime, of course, we largely lose this source of leaf surfaces, or largely lose leaf surfaces as a source of bacteria. Instead, at some of the sites, particularly Cleveland and Detroit, we start seeing a large fecal signature. And a lot of it appears to be coming from dogs, as best we can figure out based on the data. So a few caveats. One is we don't know any health effects of this. And that's actually a very difficult thing to determine. It's like, does it matter whether you're breathing in dog fecal bacteria or leaf surface associated bacteria? It's very difficult to determine that. We also don't know that these sites are unique. So believe it or not, we now have data from sites near Boulder, Colorado, but pretty much these are the only sites where we've done a detailed analysis of the bacterial communities. You'd think that people would have done this, more frequently, but so we don't know that these sites are unique. It could be that a lot of sites in the wintertime you see this large source of bacteria from dog feces. In fact, right after this paper came out, there was a paper showing that in Detroit there's something like 80,000 stray dogs or something like that. So if you think about it, it is a large potential source of bacteria. And in the wintertime, we lose this leaf surface associated source. So the total numbers of bacteria in the air go down, but a larger fraction of those appear to be coming from feces, and best we can guess, from dog feces. So there you go. Nothing against Detroit, Cleveland, but and we're currently doing more work to see if this is more of a widespread phenomenon. Okay, so let's talk about those bacteria on us and in us. The last example of biogeography. So in 2007, 2008, when we first knew that we could start sampling bacteria in lots of individual samples, so when we had this sequencing capacity, we're like, all right, what should we sample? Like, let's sample the undergrads. So they're always good test subjects, right? So we actually enrolled students in the study that were leaving an exam in general biology. So general biology has 1,500 students. They take their exam in the basketball stadium, actually. So as they were trickling out of the exam, we basically said, can we swab your hand? So can we take a cotton swab and just see what types of bacteria are living on your palm? And we use these DNA sequencing technologies to basically figure out what types of bacteria we see on there. And we ended up with something about 180 students, about equally divided between men and women. It was also an interesting sociology experiment, because some people were like, hell no, there's no way. I'm like, so it's just interesting. Some people are really curious to see what types of bugs are on their skin or what have you. Other people do not want to know at all. And there seems to be a pretty even split between those two. But anyways, what we found was pretty surprising. So again, the x-axis here is number of sequences. So think of this as the number of bacteria that we're identifying on each palm surface. So we're essentially sampling about a little under 2,500 bacteria on each of these individuals' palms. And the y-axis is, think of this as number of species. Okay, so what we notice is that women have higher diversity of bacteria on their palm than men do. So I'm not saying women have more bacteria, they just have more types of bacteria. And I have no idea why. And if you ask anybody, they probably have their own hypothesis for why this is, right? But the short answer is we don't know. For a long time, we thought this was pH related, that maybe women have less acidic skin on their palm, and that may be true. We're still trying to figure out if that shakes out. It's surprisingly difficult to measure skin pH. So we're working with L'Oreal to do this, but and this also only seems to be true for the palm and selected other skin habitats. This isn't universal across the entire skin. But perhaps more surprisingly than that is we found that only about 13% of the bacterial species found on your palm are shared between any two individuals. So the bacterial communities you have on your skin surface are very unique to you. Okay, so this started shaking out. We're like, all right, so what's the next question? Like, all right, so can we use the bacterial species you find on your skin as a means of forensic identification? And this is a study we did that was, again, just proof of concept. I'm not saying you can throw somebody in jail by looking at the bacterial communities left on the murder weapon. We're not there at all. It was more just proof of concept. And we came about this by basically the first study that suggested that this might actually work is we actually sampled the keys on keyboards of 3 individuals, the red, blue, and black individuals. And we sampled the bacteria on each of their fingertips. So again, all you need to know here is each point is a unique sample. So whether a sample of fingertips or a sample of keyboard, and points that are close together represent samples with similar bacterial communities. And as you can see, it clustered very nicely by individuals. So the bacteria found on your fingertips are very similar to those found on your keyboard. And this was after 3 days of the keyboards not having been used. And they're still remaining there, and we can actually use the bacteria left on the keyboards for forensic identification. And we did a series of other studies. So basically, the proof of concept is that you harbor very distinct communities. And in fact, identical twins still harbor very distinct skin communities. They do not change appreciably over time. So they do change, but your bacterial communities still look like your bacterial communities, regardless of when they're sampled. They remain on touched objects for up to weeks, and we can use bacteria to match touch objects to owner with more than 90% accuracy. And of course, what we didn't test is the next one is like, well, how easy is it to distinguish if multiple people touch the same object? And there's obviously lots of other studies that we have to do before this would even be close to being sort of a normal process in law enforcement. But I think what was most surprising about the study is the paper came out in 2010, and literally 2 months later, there was an episode of CSI where they were trying to solve some crime, and the scientist is like, well, I was recently reading this paper where you can use the bacteria that were on your skin. Like literally 2 months, which is impressive to me because the speed at which they work is amazing. You know, the fact that they were like writing this script after the paper came out and filming it and airing it, if only science worked that quickly. I guess if you have millions of dollars at stake, you can work fast. So what this leads us to is this finding that we harbor very unique bacterial communities on our skin. We also know this is true for mouth bacteria and the bacteria in our guts. There is no such thing as a core community. We all harbor very distinct taxa in our body habitats. So the question is why? Is it genetics? Is it parental influences? Is it your diet? Is it we'll call lifestyle, hygiene, whatever you want to call it? Is it just what you're surrounded to? Is it where you were growing up when you were a year? The short answer is we don't know, and I'm sure you could come up with all sorts of other possible factors that could influence community. Is it antibiotic usage? So forth. And this is an important question, remember, because these microbes that live on us and in us have a huge effect on our traits. And this is, by the way, this is a picture of my daughter. Here she was at about 8 months. This is a more recent picture. She's almost 2 now. And so I always think about these questions. I was like, all right, well, who's inoculating her with what types of bacteria and so forth? And I also want to point out that another great thing about having a kid, besides just they're enjoyable and cute and so forth, is you're basically growing your own test subject. And also, she's getting to be a good lab worker. So here she is in the lab, and here she is swabbing her mother's hand. Yeah, so, of course, what you don't see is immediately after this, she stuck it in her mouth. So the question is we're all very individual, so why? Why do we harbor such unique communities? And we know that it's not simply a matter of genetics. Genetics may be involved, but you can't predict what types of bacterial communities are found. In this case, we're showing data from the gut. And these are data from Pete Turnbaugh, who's now at Harvard here. But basically showed that identical twins harbor bacterial communities that are no more similar than non-identical twins. So it's not simply just a matter of genetics. And this was with gut communities, but since then they've shown this to also be true for mouth communities and skin communities. Okay, so it's not just simply a matter of genetics. And there's all sorts of other factors that we know. Again, there's studies showing antibiotic usage can profoundly alter your microbial communities, particularly if those antibiotics are administered at a young age. Also, the mode of delivery. So this is some work we did with a group at University of Puerto Rico, where we basically sampled the mothers, sampled their mouth, their vaginal community, gut community, skin community of the mothers and of the infants 2 days after they were born. And what we found is pretty striking. I know the pie charts are nearly impossible to look at. But essentially, if we look at the babies born by vaginal delivery, the bacterial communities throughout their body look like their mom's vaginal bacterial community, which makes sense. As they're passing through, they basically get inoculated with lots of bacteria. They're in the mother's vagina. In contrast, if we look at those babies delivered by a C-section, all the microbial communities on that infant look like skin bacterial communities. And interestingly, not the skin bacterial communities from the mother. And so, of course, not having, at the time, I didn't have a kid, so I was like, this doesn't make any sense. And then what of course happens is after the baby's born by a C-section, that usually gets handed to a nurse or to the father or somebody else who's not the mother, right? Because the mother is on the operating table. So it makes sense. And so basically, all of a sudden, even 2 to 3 days after birth, we see this big split between the bacterial communities on infants born via vaginal delivery versus via C-section. What we don't know is how long this effect lasts. Can you still see this 8 years after birth? We don't know. And those studies are going on right now. People are starting to do longitudinal studies to sample over time. And we also, again, don't know the effects of this. But I don't know if anyone saw, just this week there's a paper showing that babies born via C-section have a higher prevalence of obesity. Just came out just in the past few days. And they actually have some indirect evidence that it may be linked to the bacterial communities in the gut. But again, all of this work is still in its infancy, part of the putt. Okay, so another potential source, and this is data that's hot off the press. I just put this together a few days ago, or my postdoc did. And what we're also finding is that individuals sharing the same household share more similar bacterial communities, even if unrelated. Okay, so let me walk you through this. So the x-axis here is just distance. So the larger the bar, or the longer the bar, means more distinct communities. The blue bar means those from the same family or sharing the same household, and the red are just different families. So what we see is, for example, partners share more similar bacterial communities on their forehead than people not living in the same house. And obviously, we hope this is not a genetic effect, right? And we also see this with father-child, mother-child, and so forth, where basically people living in the same household share more similar bacterial communities than we'd expect by chance. And interestingly, this also works across species. So dogs living in the same house share more similar bacterial communities than we'd expect by chance. And this is not just true for the forehead, this is true for a lot of environments. And it varies depending on varies on the strength, but this is true for palm bacteria, those bacteria found in stool samples, and those on the tongue, where those individuals sharing the same household seem to have slightly share more similar bacterial communities. So the short answer is what we think is happening is they're basically exchanging microbes in their environment, or they're picking up microbes from their environment, and that's shaping the types of communities you find there. So not only do we share households and kisses and so forth, we're constantly swapping bugs back and forth. You can think about it that way. It's a little less romantic, but you get the point. So then the question is okay, so there seems to be this effect of your surroundings on the bacterial communities, on your skin, in your mouth, in your gut. What types of bacteria are in your house? And believe it or not, we have a surprisingly poor understanding of this. So we spend more than 90% of our time inside. Yet, microbial ecologists, we like to go out to these exotic locales like the rainforest and the Arboretum and so forth to study the microbes that are out there. But we know surprisingly little about the microbial ecology in your house, right? And so this sort of led us to another topic here, which is just what are the types of bugs that are out there on surfaces outside and inside our house. And again, this is very much in its infancy. We're just starting to do this work, but there's some things that are pretty surprising. So we sampled 5 different kitchens. And we just want to say, okay, well, what is the distribution of microbes around on different kitchen surfaces? So we basically swabbed every surface in a kitchen that we could get our hands on. For each kitchen, we ended up with about 150 different samples. Everything from the floor to inside the refrigerator to the oven vent and so forth. And we can start bidding these taxa based on their likely source. So we can say, okay, well, these are skin-associated taxa versus food-associated, primarily produce, so these are mostly leaf-associated taxa, and then water-associated. And we can start mapping these distributions across the kitchen. So the darker the color basically means more abundant. So not surprisingly, we see lots of skin-associated taxa on the door handles. Makes sense. Lots of food-associated taxa associated with those surfaces where we're either storing food, like in produce drawers, or on cutting surfaces or countertop surfaces, and so forth. Okay, but this is sort of just showing proof of concept that we can do this. We can start looking at bacteria on different household surfaces and seeing what's there and so forth. And none of this is particularly surprising, but this sort of gave us motivation to start the next project that's just underway. So we just started this. And here what we want to address is basically how does the type of building you live in your lifestyle, the occupancy of those houses, or the location of your house. How does all this affect the types of microbes that live inside your house or on your house? So we just recently launched this project, and it's a citizen science project. So basically, we're recruiting volunteers to swab samples from their homes, and then we're going to sort of give them, once we have results, we're going to give people the results back from their individual homes, and have them look to see what sort of patterns emerge and so forth. So all the data will be publicly available. And we just started this project basically around Christmas time, and we already have, as of basically 2 weeks ago, we have more than 6,000 households signed up. And so what we're going to have them do is sample selected locations inside and outside their house with these swabs. They send us back the swabs, and we use these DNA sequencing technologies to see what types of bacteria and fungi we find in their homes. And with the sampling of this magnitude, we can start addressing interesting questions like, if you have dogs in your house, does it affect the types of microbes living in your house? Do you have different microbes in your kitchen if you're vegetarian versus non-vegetarian? How does cleaning frequency, how does climate affect? Do you have different microbes in houses in Phoenix, Arizona versus in Boston? And these are pretty obvious questions, but we don't know the answer to them yet. And it'll be interesting to see what sort of patterns emerge from this. So again, this project is just underway. We had a lot more volunteers than we expected. So now we have to come up with the money to actually analyze all these samples. So any suggestions would be greatly appreciated. So with that, I'm going to just finish up by mentioning that I didn't do all this work myself. In fact, my students and postdocs don't even let me down in the lab anymore. And there's a lot of people at different universities that contributed to the work I presented, so I want to thank them, and of course, funding agencies. And with that, any questions? Yeah. Presumably, yes. Presumably, they're sterile until they come out. Yeah. They can't pass the placental barrier. The idea is that bacteria can't pass the placental barrier. So as far as we know, babies in utero are sterile. And yeah, obviously those are difficult studies to do, but as far as we know, they are. Yeah. So if you think about it, the infants are, as soon as they are delivered, just going to pick up. I think they're too large, so this is my guess, and I'm sure there's also a lot of immunological responses as well. But bacteria are a micron in size, so they're just too large to pass through. And we know this is true for a lot of chemicals as well. If they're too large, they won't pass through the barrier. But as far as we know, babies in utero are sterile. Yeah, and presumably it's become some sort of priority effect, like whatever hits you first. Because if you imagine there's this ripe environment for microbes to grow, so it's just whatever the first inoculum. And again, we don't know how long this effect lasts. Because this study, it's very difficult to get approval for this kind of work. And so the approval was, okay, you can sample up to 3 days, and then that's it. And of course, it'd be really great to follow up now and see, and to detect that effect, you obviously need samples collected over many years to follow and see. And it may have no effect. It may just be that after a week or two, that delivery effect is washed out. We don't know.
Given the soil pH for bacterial diversity, and given the whatever the gradient of pH is within our system from our mouth to the end of our digestive system, is there some gradient of bacterial diversity within that pH?
Within the body? Well, so in the body you have a lot of other factors besides just soil pH. Like, for example, your gut is anaerobic. There's no oxygen there. So obviously that's going to select for very different types of bacteria than we see on the skin surface, where there's plenty of oxygen available. That's our working hypothesis for diversity of skin, though, is that it may be related to pH. We don't know the answer to that yet. That we're working on that right now, but the idea is that maybe you can predict bacterial diversity by measuring skin pH. And it does, as I showed the face of my postdoc, it also varies hugely just over relatively small spatial scales. It could be due to pH, it could be due to oils, it could just be somewhat random. we don't know. But that's sort of the next step. Is okay. So the first step is now that we show these patterns, the next step is why do they exist? And then, of course, downstream of that is like, why does it matter? Does it matter if your communities are really distinct? And that's obviously a very difficult thing to unravel, but lots of people are working on that.
What's your feeling about all these antibacterial things that everybody
They make a lot of money selling them. I mean, basically, you can make a lot of money by making people paranoid, no matter what it is, right? And I don't want to give the impression that pathogens don't exist. We know there's lots of nasty bugs that are out there, and a lot of them can be acquired in hospital settings and so forth. So I don't want to give the impression that it's not important, but bugs are all around us, no matter what. So, at a certain extent, the next question is, okay, well, just obliterating every microbe is not necessarily, in some cases it may work well, but it's not necessarily always going to be the best approach. And I think there's increasing evidence for that, because we know that oftentimes if you put people on heavy doses of antibiotics, you may rid them of the pathogen in their gut, for example, but it also causes a whole slew of other problems. Because we rely on those microbes for digestion and so forth. Yeah, so I didn't show, but we actually had samples from pigs and cows in there, and we don't see those taxa that we're seeing in the atmosphere. And near all these sites, these are urban sites in these areas. There's plenty of parks around and so forth, but not necessarily animal feeding operations. And again, I want to point out that I'm not picking on these. This is just where we happen to have samples from. And it may be that this is a more widespread phenomenon in a lot of locations where in the wintertime, basically where are the bacteria going to come from? They don't appear to be coming from the lake, by the way. The bacteria we see in the air aren't at all similar to what we see in the lakes, even though these sites are bordered by are next to the Great Lakes. So the short answer is we don't know. And to do that, we just need to start collecting samples from more locations. And the EPA collects samples all the time, but they put them in the oven immediately after they're collected. So they're useless for more biological analyses, or at least microbiological analyses. Yeah. In soil, of course, there's a huge industry around basically producing the right sort of microbial inoculum to add to your soil to promote plant fertility or reduce pathogen loads and so forth. Some of it, there's probably some real science behind it. Some of it, it's a bit shady. But we know that if you manipulate the community in soil or associated with roots and so forth, whether we're talking about bacteria and fungi, you can oftentimes influence plant growth rates and so forth. Human body, we don't know, but a lot of people are working on it, right? Because if you can figure out the right types of microbes to put in your gut, then that's probiotics, right? And some of it, there is some real science that there may be some validity to it. A lot of it is shady, for lack of a better term. But there is a huge avenue of research right now. People are trying to figure out, can you inoculate your skin or your gut with sort of beneficial microbes and reduce your chance of getting pathogens. I don't know anything about Dutch-elm disease. So I think you have to ask somebody else, unfortunately.
If your surgeon had non-clinical pathogens in his mouth, would you run an operation?
That's. There is an interesting story in there. So it turns out a lot of people are carriers of MRSA, methicillin-resistant Staph aureus. So basically, antibiotic-resistant Staph aureus, common skin bacterium, but a lot of people are carriers and are completely asymptomatic. And what's unclear at this point is why some people have these bugs on them and other people don't at all. And why some people are asymptomatic and some people aren't. And the answer to that's not going to be simple. But there are a few studies that are coming out soon that are showing that may be related to the types of bugs you have on your skin that basically allow these bacteria to colonize or not your skin. So, short answer is we don't know, but a lot of people are working on that. So it'll be interesting. But it didn't quite answer your question. But
Well, all right. We're going to stop the questions. If you have more, you can come up.